I know many of you in the audience already. I've met several of you. You might be one of the farmers that I worked with on this project. But those of you that don't know me, I am a new assistant professor. I still could say new. It's just been over 2 years. And I've heard from my colleagues I could still use the new guy card for another year before I start getting in trouble for stuff. So I've got 1 more year I can use the new guy card. My official title is Assistant Professor in Soil Plant Interactions, but my background is soil fertility, soil ecology, I dabble in soil microbiology too. And soil plant interactions sounds like a fancy title, but how I explain it is there's 2 main ways that plants interact with soils. And the first way that we often think of is roots taking up nutrients and water, right? And also leaving root exudates and also sloughing off cells. But the other way that plants and soil interact are when plants die, right? And then they slowly become, well, quickly in some cases, but become soil. And that's what decomposition is, right? And so I came up with this idea to use decomposition as a cheap or inexpensive is the better term for it. So an inexpensive, low-cost, scientifically, but yet scientifically robust soil health indicator. That's really the key. That's what makes it challenging, is to have it both inexpensive and scientifically robust. How many of you were at Alan Franzluber's short course and saw he had some really compelling data? And in fact, if you were there, you might have heard me ask him, because at his last slide he said, this is an inexpensive test. And I asked him how much it was, and he hemmed and hawed a little bit, and then somebody, I think, looked up the Solvita paddles, and I think it's like $60 for 3 of them or something. It's expensive, and that's just if you want to look at the color of the paddle, let alone if you want to get one of the readers to get a more accurate value to stick the Solvita paddle in there and get an accurate reading. So this was my attempt to come up with a scientifically robust, yet low-cost method to assess your soil health. All right. So before I get started, that's just a little bit of introduction. I need to embarrass my graduate student, Teresa. She's actually sitting over here in the front. I'm going to send all my difficult questions to her. She's the one that's done all this difficult work. I get the easy job of just coming in and presenting it. And not only, I think she gets embarrassed when I show this picture. I think this is a great picture, though. It's the one she gave me to put on her lab website. But I'm also going to tell a story about her and her parents. So she actually, one time when she was collecting some of the tea bags from some of the long-term strip trials, she brought her parents with her to do some field work. And they had such a great time. And she's excited about the project and got her parents so excited about it that her dad has started his own decomposition experiment in his basement. So I thought that was awesome. And yeah, I'll have to figure out, or have to hear from Teresa how that experiment's going. Before I start, the other people I want to acknowledge, of course, are all the farmers I've worked with. Many of you were probably at James Rebank's talk, and he talked about botanists and stream ecologists coming onto his farm, and said how much he learned from them, and that it really resonated with me. But I think one thing he forgot to mention is that they were probably learning a lot from him. And that's been one of the best things about this project, is how much I've been learning from the farmers that have been participating in this and the practical farmers of Iowa. So I think that is really awesome and makes my job really exciting, and why I like what I do or love what I do. Yeah, with that said, I'll get off my soapbox and being sentimental about it. But now let's get to the brass tacks. Okay, so a little bit of an overview first. I'll talk about some background, mention these soil health tests. And of course, since this is about a low-cost or inexpensive soil health test, I'm going to give you some prices on the soil health tests that are out there. I'm going to talk about really the origin of the idea. It comes from kind of several different sources. So it's some work I did as a postdoc a couple years ago. It comes from the soil your undies thing, and like a lot of ideas, it's an amalgamation of things. And it also came from a group of researchers in Austria that started a citizen science project. And this is actually, this part, many of you of the PFI farmers out here, this is the spiel that I gave you guys, that section, to talk you into burying the tea bags on your farm and getting involved in that. So you'll recognize a lot of the slides, but many of the people in here haven't seen the rationale for it or the proof of concept. And then finally, what a lot of you are waiting for is the preliminary results, the data. And it's hot off the press or hot off the Excel sheet. Stefan, in fact, just sent me yield data about a week ago that I'll have some yield data to show and how it relates to this test towards the end of the talk. So I'm very excited about that as well as many of you are as well. And then finally, I'll wrap it up with some final thoughts and next steps. Okay, so a little bit of background. I got a screenshot from Politico. This is a good indicator of the hype and the interest behind soil health and the concern for it, I would say. And with this concern and high interest, there's a lot of interest in tests to measure soil health, right? And with this, these are kind of the reasons why you might bother to test your soil's health. It's a comprehensive assessment of soil's productivity, so that's unlike a soil nutrient test, right? Which are mostly chemical in nature, so this is biological, chemical, physical. That's what I mean by comprehensive. And it's not just about the productivity, but about the sustainability of the soil too. So how that soil relates to environmental issues like carbon sequestration in the soil and nutrient losses. We think the soil health tests relate to that. So that's one reason to do it. It also helps you evaluate land management practices, right? So a lot of these PFI farmers are trying out strip trials. I was just in a session with Stefan and some of the farmers, and they want to know, are these different strip trials having effect on their soil health? So that's a good reason to use some of these tests too, is to see what the effect of management practices are. And also can provide incentives for keeping a soil healthy. And then eventually, we're not at this point yet, but it might help assess land value, kind of like the corn suitability rating too, or the CSR2. And then the fourth reason I've come up with is because there's a lot of both public and private soil health initiatives kind of sweeping the nation. Some of the two examples that have gone through state legislators is California's Healthy Soils Action Plan, Maryland House Bill 1063. And these are more just kind of, there's not a lot of teeth behind these regulations, or I shouldn't call them regulations, these initiatives yet. They're really initiatives. And then kind of public-private partnerships like the Soil Health Institute are working to come up with soil health indicators to get at some of these same reasons that I mentioned earlier. Okay, shifting gears a little bit. So a soil health test, and these are somewhat, in my opinion, a list of the ingredients that should be in a good soil health test. So it should be based in rigorous research, one, right? I think we can all agree on that one. It should be broadly applicable across soil types and also climate types and regions. This is an issue with some traditional soil tests, like nutrient fertility tests. You need different extractions for different regions, different soil pHs. We need a soil health test that's broadly applicable. It should incorporate physical, chemical, and biological aspects of soil health, right? I mentioned that as a reason why they've gained a lot of interest. And they should have consistent, robust methods across laboratories. This is the same for traditional soil fertility tests. They should also relate to yield, right? That's the bottom line and an ultimate goal. But yield is also a biological indicator, right? That's a lot of soil health tests, and actually Soil Health Institute include yield as an indicator. So yield without fertilizer application as an indicator. So nutrient potential, or nutrient supplying potential. I'll talk about that in a little bit. And then this is the one that I tack on, is it should be relatively inexpensive again, right? We know farmers have tight margins, and I argue that this test should be cheap or inexpensive, I guess, is the appropriate word again. So this is a diagram from some colleagues of mine in the agronomy department. They came up with a soil health diagram. It includes all the physical, chemical, and biological aspects. This is really where I'm focusing, but they're all kind of connected. And organic matter kind of connects all of them, because you increase organic matter, you're going to increase some of these physical properties like bulk density, you get improved bulk density, you get better soil structure. Your soil organic matter is high in cation exchange capacity, so you're going to increase your CEC when you build soil organic matter. But soil organic matter is really related to the biological tests. And these are some of the lists. I've modified it a little bit. So potentially mineralizable carbon and nitrogen. This is what Solvita and the Haney test measure. This is what Alan Franzluber talked about yesterday and the day before. And then soil microbial biomass is another one. So just overall population, or how much biomass do you have, microbial biomass. Microbial activity. So my lab has a couple ways of measuring microbial activity. So we measure enzyme activity. So enzymes are produced by microbes to acquire nutrients and carbon. We also, another measure of activity is the potentially mineralizable carbon, so somewhat overlapping there. And then some people are talking about microbial diversity. I don't think we know enough about that to say what is healthy and unhealthy as far as diversity goes. But then the last one, and I'm going to argue for and add, is decomposition is a good indicator that should be under that biological category. And that's pretty much what this whole talk's about. And then all of these, as I mentioned earlier, should relate to yield. Okay, so who's measuring soil health? Well, these are the organizations and kind of the most common organizations that can measure. And they have a range. The reason I have a range of prices per sample is because they have different plans. Some of them have a very basic plan, and that's the cheaper end, and then they have a Cadillac plan. And the average of all of these is about $50 to $100 per sample. I find it ironic that the Cornell's test is also called cash. And it happens to have the most expensive upper limit there at $150 per sample, right? That's incredibly steep. Okay, so that's kind of the current state of things. Now I'm going to shift gears a little bit and talk about our idea. And it's not a new idea, like I said. So in fact, the earliest evidence I could find of this, I'm sure there's earlier, but this was a good quote I found. And this is from Eve Balfour. She was a founder of the organic movement. And this, I'm just going to read the quote out of her book, The Living Soil. And she said, the soil is so she had fertile there, so I'm replacing it with healthy. The soil is so healthy that it would eat almost anything thrown at it, from a gunny bag to corn stover, right? This was something that people observed in their fields before, the underwear, soil your undies thing. This was, they saw residue decomposing quickly, and they observed that it was an indicator of soil fertility, right? But I want to go back to that, and the theme of this conference is revival, and I'm reviving this idea and kind of putting a new spin on it. So that's kind of my pitch, and the pitch I gave to the farmers to get them excited about using tea bags. All right, so I'm going to go through kind of the rationale for this. The first thing I'm going to do is a little shameless self-promotion. So this is where the proof of concept came from using a soil decomposition index. I'm going to show a couple papers, and if there's something you want more details on, send me an email. I'm happy to send you PDFs of either my papers or some of these other papers that I talk about in my talk. This was a paper I published as a postdoc now 4 years ago. Crop rotation complexity regulates the decomposition of high and low residues. So what we did in this study is we took soils from 5 different crop rotations, long-term trial, 12 years, and I added 7 different residues to it to see how the history of the crop rotation affected the decomposition of the residue. So a little bit about the experiment. It was at the Kellogg Biological Station, Long-Term Ecological Research Station. This is kind of a sky view of it was a randomized complete block design with crops, the rotations in 4 blocks, replicated blocks. 12 years of treatments when I collected the soils. They all received the same tillage. They had no external inputs. So no fertilizer. This is really kind of like soil abuse. So they were just looking at how increasing diversity would increase the soil fertility. So this is a really nice gradient in soil health, right? So the M just means monoculture, so monoculture corn, monoculture soy. And then at the bottom here, and this is adapted from a paper that looked at weeds and the yield in these plots, and I'm happy to send that as well. But this is the first 5 years of the experiment, and then the months with the first letter of the month down here, and just to show you kind of the structure of the rotations. So corn, soy, oh, and I forgot to mention there were 3 cover crops, or 3 cash crops, excuse me, corn, soybean, wheat, and then 2 cover crops, red clover and cereal rye, single and then mixed. So monoculture corn, that one's easy, all red rectangles, monoculture soy, all orange, corn, soy, alternating, obviously, corn, soy, then winter wheat. Then the more complex rotation, so corn, soy, wheat with these overlapping cover crops, so the red clover here, and then the mixture of red clover and cereal rye. So there's a bit of the background. I collected soils in 2013. And then I did basically what Alan Franzluber presented on. So I brought the soils to the optimal water holding capacity, so optimum soil moisture. And then I incubated them at optimal temperature and just measured how much CO2 was produced. I was actually talking to Chris Teachout about this, doing these kind of longer incubations. And in my lab, and the way I've done this, I've always done kind of longer incubations because you can get more information and you can model the data a bit more with this. So we went out to 120 days. Some of these are 1 day, 3 day bursts of CO2, but we measured 120 days. And this shows the cumulative amount, or the overall amount that was CO2 that was produced. And we see that there's a kind of an increase. Oh, and I forgot to mention we did it at 3 dates. So spring, summer, and the fall, kind of at the end of the growing season. And across all dates, we saw an increase, but a particularly large increase in the more diverse rotations across all 3 dates. Really didn't matter which date. The other thing we could do with these incubations is we can measure nitrogen at the beginning and then at the end of the incubation. This is potentially mineralizable nitrogen. So we saw the same effect with nitrogen. So you increase crop diversity, you're increasing the amount of potentially mineralizable nitrogen. So this is the nitrogen supplying power of the soil, the organic nitrogen that can be mineralized and made available to the crops. And this actually did relate to yield as well. So in this paper, we related this to yield in these plots. Okay, so I'm going to pick out 2 soils, what I'm going to call I shouldn't say unhealthy, let's call it less healthy soil would be the monoculture corn, and then the more healthy soils, the corn, soy, wheat with 2 cover crops. All right, so this is now, that was maybe seemed a little long-winded to get to this point, but it's important just to show that one was less healthy and one was healthy. So now I'm calling this SDI as a soil decomposition index. It's a new soil health index that incorporates a difference in decomposition between 2 residues. So as I mentioned, we added actually 7 residues to all of those different soils, but I'm just going to pick out the 2 soils and 2 residues. So we added red clover, which had a C to N ratio of 13. That's what the number is in parentheses. Then we added wheat, which had a C to N of 42. Does anybody know or remember from soil fertility class what the number is or range where nitrogen mineralization occurs or immobilization occurs? I heard somebody whispering it. They actually mentioned it in the last 16 to 1, close. It's 20. It's usually a range of 20 to 30. C to N ratio 20 to 30. So So that's important here. So we have one type of residue that's below that threshold, right? 13, it has very high amount of nitrogen, meaning it has a low C to N ratio. And then we have a very high C to N ratio, meaning it has a lot less nitrogen than the other T. Or excuse me, I'm jumping ahead here. Residue. All right, so what we did is, like I said, we incubated these for actually a year and measured the amount of CO2 or how much was decomposed inside mason jars when we mixed the residue and the soil. And so what we saw is with the red clover, the less healthy and healthy soils decomposed the same amount. It was a very high amount. They're not limited by anything. But then, while I was hiding these bars, when we added the wheat, so the higher C to N ratio, we saw the healthy soil was much better at decomposing this, getting closer to that baseline than the less healthy soil, right? And some of you might be thinking, oh, that's because there's more inorganic nitrogen in this soil, right? Well, we didn't find that the case. There was no difference in ammonium and nitrate when we extracted these two soils at any points in the year when we measured it. So basically, the way this index works is very easy, is you just divide the amount that was decomposed in the more difficult to decompose residue. So 8.9, that's what this number is, divided by 12.6, which is the baseline. And then you end up with 0.71. Same thing with the healthy soil. 10.7 divided by 12.8. And you get a value of 0.84. So the idea is, the closer that value is to 1, the healthier the soil is. That's an important point we'll come back to later. Okay, so now you might be wondering, okay, so what? You showed that with a decomposition index is there. Well, not only did we show it's there, but it's related to some more traditional measures of soil health and soil biology. So the one I pulled out, it was also related to microbial biomass and a few other things, but I pulled out basal respiration because this is like the Solvita test, right? You stick the paddle in, field fresh soil, you put the lid on. This is what this is, basal respiration. So there's a lot of variation in there. It's barely significant, but it is related. And you see the kind of more diverse rotations clustering up here, and then the monoculture corn down here with the low soil decomposition index. Okay, so that's fine for a scientist like me to do, right? However, not everyone can do this. It's not easy. It was a lot of labor doing this experiment. So these were all my mason jars. I had to lug around on a cart where I mixed the soil and the residue. And then we had to sieve a lot of soil. So there's me sieving some soil back when I was a postdoc. And then it also required very expensive equipment. So we had to use an infrared gas analyzer to measure the CO2 that was produced, right? So that leads me to think, well, maybe that is a good soil health index, but it's not realistic. It's not going to go anywhere if it's not cheap or inexpensive again. But as we know, cotton is cheap, or relatively cheap, cheaper than what I just showed, and it decomposes. And in fact, I'm going back to the revival idea again. This is a screenshot of it's a compendium of research articles looking at decomposition of a cotton substrate. So this is kind of a uniform cotton substrate. Soil ecologists use this quite frequently. I was only in 3rd grade at this point, and so I've talked to some soil ecologists that are a little more long in the tooth than myself, and I haven't heard a good reason why this died out, but there are research trends that kind of come and go, and this was one that was popular at one point, but then it kind of faded away. I did find a paper, so stream ecologists are using this. They'll put this cotton in streams and look at decomposition in streams, but soil ecologists stopped using this a while ago, at least in the 90s. But it's made a comeback, right? It's been a revival. Many of you have seen the Soil Your Undies thing. In fact, we've done a bit of that research for just demonstration purposes. And what's going on with that is we're using a cotton substrate, right? That's what the underwear is. And we're putting it at the base of the food web. So we have all the microbes, actinomycetes, molds, or fungi and bacteria, usually first colonizers. We have all these shredders or communutors, like earthworms, millipedes. And then we have secondary and tertiary consumers of those. And you have this complex soil food web, but we're using something at the base of it to kind of measure the health of it, right? The idea is that the more the cotton is decomposed, the healthier the soil is. Okay, so I'm going to give you guys a little quiz here. So this is a local experiment run by Matt Liebman. He's in the audience over here. So what we did this, I think it was last summer or maybe my first summer here, so 2 summers ago. He has a 2-year corn-soy rotation with synthetic fertilizer, and then a 4-year corn, soy, oat, alfalfa rotation that receives mostly manure, but some synthetic fertilizer as well, supplemented. And we buried the underwear for 7 weeks. Which one of these, right or left, do you think decomposed more of the underwear? Right, yeah, I see a lot of people pointing to right. Yes, you'd be correct. So David Kwamenza, who's pictured there, and Madi and I went out there and dug these up. We buried it at 2 or 3 inches deep, left the elastic band out. And we had replications of this. I didn't put the standard error on it, but you get almost more than double the decomposition in the 4-year rotation with manure. Pretty cool, pretty easy, right? Another example. So this is, sorry, you have to wait till the end. A colleague of mine, Neil Sass, who works at the Soil Survey Office in Waverly, he's the one who gave me the idea of the underwear thing. He sent me this picture. So this is a demo he did. He buried the underwear in conventional corn, no-till soybeans with cereal rye cover. That one there, perennial pasture, and then the alfalfa. And those are all the percent lost weights of after, I think he did 6 weeks instead of 7 weeks. But you see a large difference, especially the alfalfa. Wow, there's nothing there but the band. Okay, a little bit about that. So you might be saying, why are you talking about tea bags then? Why isn't this talk about underwear, right? Well, it works great as a demonstration tool, but not really as, and it's cheap too, so it matches one of my 2 criteria, so inexpensive, but it's not very scientifically robust. And there's a couple reasons for that. One, it's messy and inconvenient to retrieve the underwear, so we had cotton peeling off and sticking to aggregates, so we know we were missing some of it. We had a lot of issues with that. And then another thing is that I didn't show here, but we buried a pair of underwear in a native prairie, and we thought, oh man, it's going to look like the alfalfa. It's going to be nothing but the band. But it turned out that underwear hardly was decomposed, and it was just because that soil was a little bit wetter. There were crop fields next to it, but it was wetter. And so things like temperature and moisture are stronger regulators, I think, than the biology in the case of that. So that adds a lot of variability. This is also kind of the problem with the Haney and Solvita as well. And it's also and makes it difficult to compare among treatments, right? So the question that leaves us, or left me with the question of, can we develop a more scientifically robust soil health indicator, but yet is still relatively inexpensive? So now we have arrived at the tea bags finally. So these are a group of researchers from Austria that published this paper around the same time I published my postdoc paper. And they were looking at tea bags as a way to measure decomposition, mostly in natural systems, so grasslands, forests. They even had some tea buried in Antarctica, I think. I went to their conference, actually, and met with these folks. And they wanted to do citizen science, and they wanted to do education, but I've been trying to convince them that we need to use it as a soil health index, and I think I'm getting them to come along with me. And they have a website, too, and I can send it to you if you send me an email, where they're getting citizen science data from around the world, including farm data. And I've submitted data from your farms to them as well, those of you that have participated in the study. So this is what they did. So I'm borrowing a little bit of data in the paper and just kind of making the case for it as a soil health index. So these were some data that I'm basically just adapting in graphical form to make it easier to show you guys, to translate it to soil health index. So they took these T's, a red T, rooibos T, and green T, and if you notice, the C to N ratios are remarkably similar to those residues that I used, right? So my wheat residue was 42. This rooibos tea, which is from South Africa, it's a bush tea, is 43. And the green tea, which has a C to N of 12, was similar to my red clover residue that I added to the soil, which was 13. Remarkably similar, just total coincidence. I hadn't met these people yet. And what they did is they decompose at different temperatures. So we know we get this range with temperature and moisture, right? That was the issue with the underwear, right? So, and it goes up and down about the same amount, at least when they did 60 and 80 degrees here, each T. So, sorry, I forgot to mention, they did it for 0 to 125 days, and this is the mass remaining. And then the average is kind of this dotted line in there between 60 and 80 degree Fahrenheit. Those are the outer boundaries there. So let's say that's our less healthy soil. So going back to the soil decomposition index, we'd expect a healthy soil, or hypothesize a healthy soil, should draw down or decompose quicker this rooibos tea, right? And then we can calculate an index, that same decomposition index that I proposed, and I showed an example from my proof of concept in my paper. We can calculate that on the healthy and less healthy soils. So I did that with their kind of data and this kind of example of a healthy soil. So if we have to subtract 100 minus 65, so the red is the rooibos tea, the green is the green tea, and I forgot to mention that the shape of these curves. So the green tea decomposes very quickly and has this very rapid and fast drawdown to a plateau because of the low C to N, or high quality. It has a lot of nitrogen in it. And the rooibos looks like that because it has a higher C to N, less nitrogen in it. Okay, going back to the index. So I did this, and so you end up with 0.50 in this hypothetical example, or 0.5, and then 0.79 for the healthier soil. And again, the idea is that the healthier soil should be closer to 1. Same idea as the residue study I showed. Okay, so I know I'm setting this up a lot, but it's important. So the benefits of using this SDI with 2 T's, as opposed to maybe using the underwear. One, it's very inexpensive. So it costs about $20 for 60 pyramids. Those of you after my talk, if you want to see the pyramids, so I brought a couple boxes up here. We have a large stock of these in my lab. So with those 20 or 60 pyramids, you can bury those in 30 locations around your field. So you just have to pair the green and the red tea, right? The tea is a convenient litter bag. So one thing that I didn't mention yet, but it's one of the benefits, is that this bag is actually nylon and doesn't decompose. That helps us get the mass of the residue back, or the tea back. Unlike that was the problem with the underwear, is that some of the cotton was peeling off on aggregates and we were losing some of it. And that's actually a litter bag. Why I put that in quotes is what soil ecologists use to study decomposition. So often we make it out of screen door mesh, different size mesh, and we'll put a litter out in a field and then let it decompose. So it's an already convenient litter bag. And then this is the real key. Using the 2 T's with the green tea as the baseline, this might correct for any temperature or moisture variability. We don't know this for sure, but at least there's some preliminary evidence that this seems to be the case. And some of that is from this paper where they incubated at 60 and 80 degrees. And it seemed to increase the decomposition about the same amount. So if we're comparing things that change the same to temperature or moisture, then we can correct for any temperature or moisture differences. That's the real beauty of this. Okay, the last thing on that slide, and it's an integrated measure of soil biology. And not only is it, so I mean integrated over time, right? So we leave the residue in the field, so it's incorporating temporal variation over the growing season too, whereas a lot of the tests, including the Haney test, the CO2 burst test, you grab a soil and it's a snapshot at one point in time, right? Where this is more integrated over a growing season. And yeah, and it's, but like the Haney test, it's also it's kind of a holistic measure of soil biological activity. So I can guarantee this probably relates to soil microbial biomass, but also the resources available to the microbes, the labile carbon, the potentially mineralizable carbon, and nitrogen available in the soil. And here's kind of, I'm not an artist. This is my rendering of how that kind of works. So we might have the less healthy soil and healthy soil again. They might have differences in organic matter, total organic matter, right? Total carbon and total nitrogen. This is very hard to detect. Our equipment to measure this as scientists and the variability makes this very hard to detect. But they also have different amounts of this labile carbon and nutrients. And those of you that saw Alan Franzluber's talk, this is what he was measuring in that. And so the biology, the soil biology are a part of this pool, but also they utilize it to break down the residue and difficult to decompose residue, like maybe a very high C to N ratio corn litter. And then invariably, so I've been giving a bunch of extension talks around the state of Iowa since my colleague Madi Al-Casey's been out of town. He's on sabbatical. And I often get asked by farmers at these extension events, well, don't we want there to be less decomposition, right? That means there's more soil organic matter, right? If we have less decomposition, there's less carbon leaving the system. But this is just because we're looking at initial stages of decomposition when we can still see the residue that's there. If we were to go out later in decomposition, when you couldn't tell really any difference between the CO2, a larger and more efficient microbial community, the soil biology, would stabilize more organic matter. So it kind of creates this feedback where the biology's adding more stabilized soil organic matter, increasing this labile nutrients, and that just creates a positive feedback. But that is a good question. I get that question a lot. So I just wanted to head that off at the pass. Okay, so this might sound great and grand, right? But I recognize there's limitations with this method, right? There are some limitations. One, it doesn't measure those larger soil fauna, right? I showed that soil food web. And the mesh size on these tea bags, the nylon mesh, is about 1/4 of a millimeter. So you're excluding a lot of the soil biology. But, however, I think that the measuring the kind of a base of that food web, so the fungi, the bacteria, the actinomycetes, even smaller soil animals like protists, measuring their activity is a good indicator of the rest of the food chain, right? So here's what that looks like. So I used the underwear here before as the base of the food web. But now, this is really who we're measuring, right? We're assessing these guys and some of these smaller protists. But we're not getting any of the shredders. We're not getting any of these other higher level consumers, really, with the tea bags. So that's one limitation. The other one, and I hate to get mad at a company for doing the environmentally correct thing, but they messed up my experiment, right? So they used to make these tea bags in nylon, so it was not very decomposable. But they've switched, and if you want to see these after my talk, you can come down here. They've switched to a decomposable one. That, yeah, that's great. They're doing the environmentally correct thing there, but it's messed up the science. However, I have another caveat to this, is that some of those folks I mentioned in Austria, they've been doing some tests, and they find that it doesn't matter much. Really, it's the tea in there that decomposes. The bag decomposes a little bit, but they can correct for it. So there's some good news there. And they've done some side-by-side trials with the old tea bags and the new tea bags. So that's another limitation. The other one is that rooibos tea is not easily available in the US. Raise your hand if you've had rooibos tea before. Okay, man, you guys are fancy tea drinkers. I guess you know where to get your rooibos tea. I looked around in Walmart. So I tried to look. There are places to find it. I couldn't find this Lipton one. But I couldn't find it in Walmart. I couldn't find it at Wheatsfields. I couldn't find it anywhere, this brand. So that's another limitation. So if you want to do this study, you have to come to your local tea dealer, which the PFI farmers have been coming to me. I order it overseas from a distributor, a Dutch distributor. Then another limitation is the effects of temperature and moisture may still make the comparison difficult, like other biological tests. But we don't know this yet. It's worth finding out, which is why we often take temperature and moisture measurements when we're out making these tea bag decomposition measurements. All right, so that's a bit of my justification. Hopefully I've convinced you guys, like I did with the PFI farmers, that this seemed like a good idea. When I gave this that same spiel to them, what was that, Stefan, like a year and a half ago? They were all excited. They're ready to order pallets of tea right off the bat, right? And their excitement, it really rubbed off on me as well, and I got really pumped about it. So we had a project funded from the Leopold Center. I don't even think we knew we were funded at this point yet. We just thought we're going to do it anyways. So luckily we got funding from the Leopold Center of Sustainable Agriculture. And we had 10 replicated strip trials through PFI. So these range from farmers doing strips of cover and no cover for the first year, all the way up to 15 years of cover and no cover. And then we have some more citizen science. So it's a range of citizen science, I like to think of it. So this range is more scientist controlled, or scientists in quotes, meaning my grad student and I and Stefan. And then more farmer-collected data. So this is getting closer to the citizen science side, where I'd like to see things going, democratizing science. So we kind of split it up in those two ways. So these red ones are where all those replicated strip trials are. Those are the farmers out in the audience. You probably recognize where your spots are. Not only did we decompose tea bags across 130 days out in the field, but we collected 2 samples, one at the beginning of burying the tea bags, and then one at the end when we collected the last tea bag. So that was in October, typically. We measured soil temperature and moisture because we know that's an important factor in just the rate of decomposition. And then we took some other soil health measurements, including physical ones. We're going to do some of the biological tests, like potentially mineralizable carbon, microbial biomass, some of these other things to compare to the tea bag index. And then here's a picture. I got this from the PFI website of what the strip trials look like, so cover, no cover. And then in the farmer-collected data, we had voluntary soil samples that they wanted to send in. And we started with 10, but we ended up with 6 that sent us tea bags. And then we had them record observations. So what the crops look like, soils, conditions, et cetera. All right, so I'm going to start with the graduate student and myself collected data, mostly Teresa collecting this. So I have different farms here. I'm not going to go through this whole table, but we had 9 out of the 10 farms. We lost 1 farm, and I'll show some pictures of what happened to farm number 10. But we have A through I. We have a range of years in cover crop, as I mentioned, from 1, a couple in 8 and 9 range, and then a 15. I will mention, so I've tried to keep these anonymous. Humans, by their nature, are competitive, but I think farmers are even more competitive. So I'm not using any names here, but I'm sure you'll recognize maybe a few of your fellow PFI farmers' farms, and you'll find your own, of course. But I will say that Farm B was the Iowa State Soil Tilth Lab, NLAE run farm, so Tom Casper's farm. What else did I want to say? Oh, yes. So we had a variety of soils, and we had a variety of crops, majority of corn, but we did have 3 sites that had soybean, and then a wide variety of management practices, including a few we haven't got yet. If you see your farm up there and there's a question mark, come see me. I'd like to fill in this table. And yeah, that's all I'll say about that table. Now I want to talk a bit about the methods. So we adapted that TBAG, the citizen science paper, and what we adapted it for row crops. So this is again, I'm not an artist. I have these very poor drawings in PowerPoint. So this is a 30-inch row, 2 rows of corn. We tried to keep the tea bags about equidistant from each other and from the corn rows. So about 10 inches in each row of tea bags, and then we alternated tea colors. So red, green, green, red, red, green, green, red. You get the idea. We had a total of 6 out there. We collected them. We buried them between May and June. So this just corresponded to kind of when management was done for the growing season typically, so some of them were in June if they were doing split application. And then we retrieved the tea bags at these days, so 4, 7, 14, 30, 68, and 130 days. And we dried them. We had volunteer farmers dry their own out of the sun, but in a dry place for a few days before sending them in. And then we just simply weighed the mass loss, because I forgot to mention we weighed them before they went out, so we know what we started with, and then we weighed the mass loss at the end. All right, so I'd mentioned how I lost one site. So this is one of the farms. This is a frustration of every farmer. You get a critter in your field chomping on corn. Well, it's a frustration for soil scientists as well. We had a critter digging up the tea bags. Sometimes they just leave them on the surface, and we'd get there and we'd rebury the tea bag. Sometimes they'd rip them open, and it looks like ate all the tea. But to add insult to injury, they would then crap in the hole that they dug the tea bag out of. So this was, we had one site where it was just totally decimated, so we had to get rid of one site. But 9 out of 10, I feel like is pretty good, especially talking to some of these colleagues in Austria that they've said they've lost about 50% of their tea bags sometimes. Okay, so now I'm going to show a lot of graphs that look like this. So I'm going to walk through this one kind of slowly. So we've got day on the x-axis, so 0 to 140 days, and then the mass loss. You've already kind of gotten used to looking at that, right? So I have a scale from 20 to 100%. So this is when we start at 100% in the decomposition curve. And then, so the green with the white fill is the no cover crop, and then I put green in parentheses. Cover crop is the filled circle, and then the red. And if you're colorblind, this was a poor choice of colors, but it just happens to be the color of the tea. But the least you could do is just see that the red tea is always least decomposed, and you could tell a square from a circle. So that's what to look for if you do happen to be colorblind. I apologize for that. Okay. Oh, I just wanted to mention that. So this was Farm B. This was Tom Casper's long-term cover crop trial, and there's some variability. These are the means shown here, and these bars are the standard errors. So there's quite a bit of overlap. It doesn't look like there's much of a treatment effect there. We haven't done much stats on these yet. And now here's all the farms. So farm A to farm I. You see a lot of variability. There's no, at least really sticking out to me, there's no treatment effect. You can see a T effect, of course. And then one thing that we did notice, and maybe some of you astute audience members out there might have noticed, is that at about our last tea bag, so the 130-day one, the mass actually went up in some of the plots. So we have a couple theories for why this might be. One, soil can get into these and add to the mass, so there's a way to correct for this. It's just burning off all of the organic matter, and then you could subtract out any mineral stuff in there, which will be the soil weight. That's called ash-free dry mass. So we could get that and correct for this. The other thing it might be that we're going to have a more difficult time is if there were some fine roots growing inside the tea bag, and that was adding to the mass at that time point. So that is going to be more difficult to tease out. And we did find some of these farms that had roots growing out like this, and we took notes and we did our best to kind of excise the roots out, but we might have lost some fine roots inside the tea bag, which might have increased some of the mass in those last dates. Which is why, but the other dates look good. They look like a normal decomposition curve, which is why I'm really going to focus on this day 68 for the decomposition index. Okay, so now here's the big summary slide. This 7 out of the 9 farms did have a higher SDI. Well, look, before I get to that, as you'll know, there's not much bragging rights here, right? So you guys are already the cream of the crop. So I think they're, even your no-cover soils would have a high SDI, or decomposition index. What I need you guys to do next year is go into your neighbor's yard and bury some tea bags at night so we could get kind of the lower values here. So everybody's kind of at a same level, but you will notice that 7 out of the 9 farms marked with these stars here did have, so the cover crop is in the blue. I have the farms on the x-axis and the decomposition index of 0 to 1. Closer to 1 is better. 7 out of 9 showed a higher mean. If we correct for landscape position, meaning we pair the 2 strips that were right next to each other, so a lot of these farms had strips going from high landscape position down to low to incorporate that variability. I didn't show the graph because it gets kind of complicated, but if we do incorporate that, then it does actually improve our data a little bit more. But even if we don't, before I get to that, the other thing that's interesting about this is that it doesn't matter whether you've had cover crops for 15 years or it's your first year doing it. It seems like there's no trend. So if we took Tom Casper's farm, the one that is affiliated with Iowa State, this should be the highest bar, right? But it's not. We see some of these one-year bars that are quite high in the cover crop. So it definitely doesn't matter how long you've had cover crop. It seems like the benefits in 1 year. And this might relate to a quick change in microbial community, maybe a quick change in available resources. The other thing I was going to mention is that when we take the overall average, we do see a significant effect, an increase from adding cover crop across all of the farms. That's pretty neat. Okay, this is even better. So this is hot off the press. Stefan hasn't seen this yet. I think I might have mentioned it to him. He sent me the yield data, as I said, a week ago. And what I did is on the x-axis here, so this is the change in the decomposition index in the cover crop versus subtracted from the no cover crop. So if you look at this kind of faint white line, if you move to the right of this, your cover crop plot increased your soil decomposition index. If you move to the left of this, your cover crops decrease that decomposition index. And then on the y-axis is the percent change in yield. And I put percent change in yield because I've plotted in here both corn and soy yields for that year. And so one thing you'll maybe notice at first, it looks like just a scatter. There's one site in particular, one farm in particular had very high increase in their cover crop strips versus the others, but had a low soil decomposition index. So I don't know what's going on with there. Maybe Stefan swapped the cover, no cover yield on me. We got to look into the management on this site as well. So that might explain this. This was actually one of the ones that was negative here. So it's actually site I here. But if we remove that one, you might notice a trend, right? Greater soil decomposition index, more yield. that you get in that cover crop plot. Lower, you get a decrease in yield, or yield drag as people often refer to it, right? Close to 10% here. So it does somewhat relate. This is really fascinating. I think this reflects that it's probably, this index is probably related to resources. Maybe nitrogen, maybe some other limiting nutrients, but it's reason to further pursue this at least. All right, so that I'm going to shift gears a bit. That ends all the strip trials, the work that Teresa has been doing, spending a lot of her time. But she's also been working with these citizen science, or what we're calling the teabag kits. So Stefan and Teresa and I sent these out to 10 farmers. We only got 6 farmers that sent us the tea bags back. 7, actually. There's one caveat. One was from California. So this is a picture of one of the kits when they came back. And I think this is really cool, right? So we've got farmers, many of them in this room, probably many of the 6 that sent these in. These are busy people, right? You know, they're running a farm, they have family, don't have a lot of free time, yet this speaks volumes of how much they care about soil health. To be out there spending their time burying tea bags, digging them up, taking excellent notes, some of the best scientists I've seen. So I think that's awesome. I think that's really cool. Okay, so anyways, so now we're going to look at some of the farmer collected data. These aren't replicated, and they're also not always cover, no cover. So there's kind of a smorgasbord of various treatments. Okay, so here, there were a few that had cover and no cover, and now I'm calling them, instead of lettering, I've got farm 1, farm 2, farm 3, farm 4. So there are 4 that had, and like I said, many of them are not replicated, but one of them did have replicates. And same kind of thing, there's a lot of noise, maybe some treatment effects, but you can't tell again because it's not replicated. I picked out 68 day because, again, some of these went up a little bit in mass, and also that's what we used for the last one. Here's farm 1 through 4. One of them increased. The other 3 showed a decrease and no change in the average when you average those out with the farmer. Oh, the other thing I forgot to mention is the tea bag kits were also the new type of tea because we were running out of the old tea. So we had the farmers use the newer teas, the one we knew that if we make more tea bag kits in the future, those are the ones we're going to use. Okay, so here's farm 1. So they had a no cover and cover, but they also had a cover and grazing. And then they had pasture treatment as well. So this was the one that did show the increase with the cover crop. They saw a decrease with grazing. And then pasture was kind of in the middle or close to the no cover crop. Okay, farm 5. This is a very ambitious farmer. So he had low, medium, and high productivity, and then a grazed pasture and alfalfa. And hearing these treatments, you probably know who they are. But we had a very counterintuitive gradient here, right? So I would have expected the low productivity to be this value and then go up, have a higher decomposition index as you got to higher productivity. The grazed pasture and alfalfa are higher, well, the alfalfa is the same, but the grazed pasture is higher than the crop. Farm 6, they compared corn-soy rotation with alfalfa. They saw a little bump in alfalfa when we took, so they didn't have the final day anyway, so this was the only value we had when we calculated the index. And so I mentioned one farmer, he's a younger farmer from California, and I connected with him because he's interested in becoming a graduate student in my lab. And he got excited about the project, and so I gave him some tea bags. He works on an educational vegetable farm back in California. And he had some plots where he was adding biochar. So one where he's had long-term biochar additions. We did see a little bump, but again, we don't have stats on it. I'm just kind of showing you guys values here. So now to kind of wrap things up and assess where we're at and some final thoughts. This decomposition index, in my opinion, does show some promise, but we've got a lot of work to do still. So there's more data analysis. So Teresa's going to be very busy. She's got more soils to analyze, still finishing up ashing the tea bags. But we want to look into further ways of measuring this too. Maybe it's not just picking 68-day, but looking at the rate of decomposition, which is why we did multiple extractions. And then we want to look at other dates too. Maybe it's not 68 days, but the 1 month, maybe just leaving it out 2 weeks is valuable. And then we want to look at other data, explain the variation, and including management practices that might explain some of these unexpected results. Climate, we've measured soil temperature and moisture at a lot of these farms. And then look at other soil factors and soil measurements. We want to validate with more traditional soil biology tests, so microbial biomass, CO2, potentially mineralizable nitrogen. And then expand to other experiments in Iowa and beyond. And this is including, I'm trying to talk the Austrians into sharing or using my idea on their farmer data set, which they've got hundreds of farms in Europe and around the world that have those same data. So I think it would be really cool if they could do that. Still trying to convince them. If you're a farmer in the audience and you want to do this next year, contact me. We have extra tea bags, and we're going to order some more for our next round this year. And this is just my last kind of final thought. I really like citizen science, or farmer science in this case, and I think it can lead us to a greater understanding of soil health and increase the adoption of conservation practices. I say that kind of at my behest because I realize this puts me out of a job, right? When you democratize science, you're putting the scientists out of a job. But it's not really like that, right? I mean, I still can work with these farmers and consult and help interpret data. And I really like seeing people collecting data and getting excited about science like I do. And just to show what it would look like in the future, or all we would need is to have a set of the tea bags, and then a scale, their own scale. And you need a way to dry the tea down well, and a lot of ovens don't get to the lower temperature we need where you would lose plant matter. So a lot of ovens are a little bit too hot, even at their lowest setting, basically, to dry these tea bags down. But in essence, this could cost $32 to be assessing your soil health very frequently, too, and at a larger scale that you could do with any other test out there. All right, with that, I'd just like to throw a few more acknowledgments up here. The people I've collaborated with at Iowa State. We had a few connections with Iowa Soybean Association. Some of the farms on there were affiliated with Iowa Soybean Association. Of course, Practical Farmers of Iowa. Stefan has been a huge help. Sarah, we've sat over a lot of cups of coffee, beers, talking about this stuff. And then the farmers that allowed us to bury tea bags, collect soils, come on the farm. And then finally, I'd like to put a plug in for the funding, which came from the Leopold Center for Sustainable Agriculture. And at this point, that's my email, and you can follow me on Twitter and any kind of updates on the tea bag on that, so soil-plant interactions, and email me if you want to get involved with this. And at this time, I'd like to take some questions, if I have time. Did I go over? Yes?
How would an ordinary food dehydrator work?
That's a great question. So he asked, how would an ordinary food dryer work? That actually might be a good, if you have a food dehydrator, that might get the moisture low enough that you could get a good reading. I don't know if anyone's done that yet. I have looked at papers, actually Stefan sent these to me, people using microwaves to dry down plant matter. I don't know if the microwaves would melt the nylon or have some issues with drying it out, but it's been used for plant matter, and the tea is plant matter. So that's one potential we're looking into. Yes, there in the middle. Good question. So he asked, do you think our results were compromised by the fact it was a drier year? Of course, that's one thing as agronomists or soil scientists, it's always better to have more years of data. You're absolutely right. So having one, we don't know if it was the climate, maybe it could have been other issues that were particular to that year, but that's why we're doing it again this year. Yes, sir. We haven't done that yet. So he asked if there were a lot of different variations in the fertilizer and other management factors. We haven't teased those apart yet with those strip trials, but that's something that we need to do to see. It might explain that one outlier in the relationship with yield. Good question. Yes? I would recommend 100th place. So 2 decimal places on balance at grams. And that one I showed is $12 on Amazon. And it has the accuracy needed. Yes? That's a very good question. So she asked if there's any variation in the T from box to box. So that Austrian group has looked at this, and they found even the chemical differences very, very little. And you'd expect this from a corporation, right? They have QA, QC. They want to make sure their weights are right. But that's why we weigh every bag before it goes out anyways. But people have looked into that, and they're very consistent. That's one of the reasons why it makes a nice measure as well. Yes, in the back there. So that's $12 for one paddle? Okay. Which lab is that at? Woods Hole. Okay, that's a great deal then. That's for the whole package. That includes the physical and chemical. Yeah, I know that the prices I showed include those other things. Yeah, you're right. So sure. Yeah. Yeah, it would work. I would recommend doing both if you can. If you're going out there to take a sample, it'd be great. In fact, I've been talking to Alan about him doing CO2 burst and burying tea bags. That's what we're trying to do as well, to validate it. Mm-hmm. That was after 12 years. So both, yeah. No, not at when the experiment was first started. I think you're asking, was there one done right when the experiment was initiated or before? No, no one did that. To see how it changed over time. I think that's what you were getting at.
All right, I'm going to call time. Let's thank Marshall for his presentation, for coming down.
Thanks, guys.